Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Kerala SET Biotechnology question paper conducted in the year January 2024. If you are new to my channel, please do like, share and subscribe. If you want previous year question papers of Kerala SET Biotechnology been answered, you can go to my channel Omni Bio Entrance, go to the playlist. In the playlist, select for Kerala SET exams. In that you will find. Coming to the first question. Which of the following is not true about type 2 restriction endonuclease? Here cleavage and methylation are by different enzyme. Yes, cleavage is by restriction endonucleases and methylation is by methyl transferases. So that statement is correct. They have asked for not true. In this case, the recognition sequence is asymmetric. No. Recognition sequence is palindromic. That means it is symmetric. It is symmetric recognition sequence. So that statement is wrong and that is your answer. It cleaves within the recognition sequence. Yes. It can't cleave the methylated sequence. That is also correct. Then next one. Which of the following enzyme is a neo-isochisomer? So it is a restriction enzyme that recognizes the same DNA sequence but have different cleavage site. See, in the case of SMA1 and SMA1, the sequence is GGG-CCC in both, but they cleave at different sites. So they are neo-isochisomers. So the answer here is option C. Then the endonuclease HA3 and SAU3A recognize dash sequence. So they recognize HA3 recognize GGCC, it is 4 nucleotide, and SAU3A it recognizes GATC. It is also 4 nucleotide. So it is a tetranucleotide sequence that they recognize. So answer here is option B. The various steps for the construction of libraries are. So first one will be isolation of genomic DNA. Then comes fragmentation of the DNA. Then comes vector preparation. Then comes ligation and introduction to the host. Then amplification. So it is 2, 1, 5, 4, 3. 2, 1, 5, 4, 3 is in option B. So option B is the answer. Then next one. Choose the incorrect statement for oligo-DT cellulose. Now oligo-DT cellulose, it is used for the separation of mRNA from other species of RNA. It is DT, that is thymine is attached to the cellulose. So the mRNA, it will, with its poly A tail present in the mRNA, it will bind to the T, oligo-DT cellulose. So it will bind to T. A will bind to T through hydrogen bonds. So the first statement, it is used for the separation of polyadenylated mRNA from other species of RNA. That is correct. Oligo-DT are covalently attached to the solid support via OH bonds. Yes, that is also correct. A solution containing RNA is passed through the solution. Yes, a solution containing mRNA, tRNA, rRNA, all three combined are passed through the column. Poly A tail attaches to the oligo-DT by ionic bonds. It is not ionic bonds, it is hydrogen bonds. So that is the incorrect statement. So your answer is option D. Then next one. Luciferase genes are used as reporter genes for screening. Choose the correct statement about them. They are obtained from fireflies only. This only word coming in your statement, you have to be careful. Most of the time it is a wrong statement. Okay, so be careful when only comes in the statement. They are obtained from fireflies. It can be obtained from fireflies, glowworms, sea pansy, all these. Then the detection requires the provision of substrate which produces light. Yes, that's correct. Here what happens is the substrate used is D-luciferin, and it also requires ATP and oxygen. So the detection requires provision of substrate and which produces light. That is correct. Enzymes such as beta-galactosidase require substrate X-gluc. It is not X-gluc, it is X-gal to produce light. They don't produce light. Beta-galactosidase, they produce a colored precipitate. So C is not correct. Luciferase genes are preferred over fluorescent protein. That we cannot say accurately because they have their own disadvantages and advantages. Luciferase genes and fluorescent proteins. So we cannot say it 100% that this is good or 100% this is bad. We cannot say like that. So D is also not correct. Then asymmetric PCR differs from normal PCR in. So asymmetric PCR, one of the primer is always used in higher concentration than the other primer. It will be in the ratio 10:1 or even 100:1. Whereas in normal PCR, you use equal primer concentration. Then asymmetric PCR, it is used to generate single-stranded DNA for sequencing, probe generation, mutagenesis studies. Whereas normal PCR, double-stranded DNA is the product, amplified product. So asymmetric PCR differs from normal PCR in range of annealing temperatures. No. The amount of primer used. Yes. B is the answer. Then which of the following is obtained via cDNA clone? ESTs. Yes, that is obtained via cDNA clone. It is express sequence tags. They are molecular markers synthesized from sequencing of random cDNA clones. Then next one. GFP is one of the marker which is used for screening libraries in hosts other than E. coli. Choose the incorrect statement for GFP. Now GFP, it produces blue light. Okay, now how does it produce blue light? By first it will be excitation of blue light. Sorry, it gives out green light. Okay, not blue light, it gives out green light. First will be excitation of the blue light, then energy transfer to the GFP's chromophore, and from there emission of green light will take place. This is how GFP is working. It stands for green fluorescent protein. That's correct. It is obtained from bioluminescent jellyfish and produces protein aquarin which emits blue light. Yes, that's also correct. The absorbed blue light produces green light which can be detected further. Yes, that is also correct. The blue light is produced because of binding of sodium ions. That is the incorrect statement and that is your answer. I already told you that in GFP, the excitation light is the blue light. That excited energy will be transferred to the GFP's chromophore. From the chromophore, the light emitted will be, after excitation, will be green light. That is how GFP is producing its green color. Then next one. The common gene delivery system for in vivo gene therapy is. So in vivo gene therapy, it involves directly delivering your gene of interest into the body of the deceased patient. It is usually involves use of vectors for gene delivery. So first what happens here is if you have a non-functional gene in your body and you want to replace it with a functional gene, you first package the functional gene into the vector, then introduce that into the patient's body. The functional gene will attack the target cells, and there it will either replace or deactivate the disease causing gene. That is what is happening in vivo gene therapy. So your answer is adenoviral vectors. Then which among the following is a human amnion derived cell line? HeLa is derived from cervical cancer cell line. L is derived from mouse fibroblast cell line. MRC5 is from human lung fibroblast cell line. So what is remaining? B, wish. It is from human amnion derived cell line. Then next one. Which of the following statement is incorrect for culture and maintenance of mammalian cells in vitro? Transformed cell lines need an exogenous supply of serum for growth. That's correct. The cells that are obtained directly from an organism is primary culture. Yes. After dissection of the organism, you take the cells, then you place it in a suitable culture medium and allow it to grow, then it is primary culture. Trypsin is added to cell culture dish to maintain cell viability and health. No, that is not correct. So that's your answer. It is not cell viability and health. It is used to dissociate the cells from the culture surface. Okay, that is why you use trypsin. And what it will do is it will break down the cell to cell and cell matrix interaction. It is usually used in subculture or in passaging of the cells. Then HEPS is generally used for pH balance in animal cell culture media. That's also correct. All the following statements regarding RFLP and RAPD are true except RAPD is quick method compared to RFLP. Yes. RFLP is more reliable than RAPD. No. Oh, sorry. RFLP is more reliable. Yes. RFLP is more reliable than RAPD. That's correct. Species specific primers are required for RAPD. No, you require random primers, not species specific primers. Random short primers are required for RAPD. Markers, but not species specific primers. Radioactive probes are not required in RAPD. Yes, that is also correct. Then in plant tissue culture system, what is the function of elicitor? Now what is an elicitor? It is a compound which in small concentration can activate different plant responses. Okay, such as defense response, production of secondary metabolites. All these can be the function responses that an elicitor elicit in a plant tissue culture system. And elicitors can be biological, that is fungal cell extract, or they can be chemical such as jasmonic acid, or physical agents such as light, temperature, radiation, etc. So induce cell division and callus formation? No. Stimulate production of secondary metabolites? Yes. B is the answer. Induce the formation of somatic embryoids? No. Induce soma clonal variation? That's also not correct. Then next one. During recombinant insulin synthesis, the bond between insulin and pro-insulin polypeptide and galactosidase can be removed by using. So insulin, you know, it is a dimer of A chain and B chain which are linked together by disulfide bonds. Now what is this recombinant insulin synthesis? So in that case there is a vector, here also another vector. In this vector you introduce the A chain, insulin A chain, and in this one you introduce the B chain, and that is fused with the beta galactosidase enzyme in both cases. Now you transform the E. coli, select using ampicillin resistance gene, then you purify the protein, that is insulin A chain fused with beta galactosidase and insulin B chain fused with beta galactosidase, you purify. Now what you have to do is you have to remove the beta galactosidase after purification. So what you do is you add the chemical CNBr or cyanogen bromide, which will cleave the A chain from the beta galactosidase and B chain from the beta galactosidase. Now you purify both A and B chains, then you treat with chemicals to combine the polypeptide to produce functional insulin. That is what is happening in recombinant insulin synthesis. Now the question is, the bond between insulin polypeptide and galactosidase can be removed by using. That is over here. After this purification step, the bond between insulin polypeptide and galactosidase can be removed using cyanogen bromide. Answer is option A. Then freedom 2, the first virus resistant transgenic crops of a transgenic squash variety commercialized in 1995. It carries gene coding for. So it carries two genes, that is the code protein of zucchini yellow mosaic virus and code protein of watermelon mosaic virus. So answer here is viral code protein genes. Answer is option C. Then next one. Identify the correct statement on nitrogenase enzyme used for nitrogen fixation. Nitrogenous mediated nitrogen fixation is energetically expensive process because it requires 8 ATP molecules per nitrogen reduced. So it is an energetically expensive process. The gene coding for nitrogenase is under a constitutive promoter. The promoter is usually responsive to nitrogen availability. So it is not a constitutive promoter. Constitutive means continuously been encoding the gene nitrogenase. So it is not the case with constitutive promoter. So 2 is not correct. Nitrogenase is highly sensitive to oxygen. Yes, that's also correct. Endogenous availability of cofactors of nitrogenase is very low. That you cannot say because the cofactors of nitrogenase are iron and molybdenum and they are present in nitrogenase enzyme itself. So fourth one cannot be the right answer. So 1 and 3, so answer is B. Then, which type of therapy was introduced for the first time in 1990 on a 4-year-old girl with ADA deficiency? So, ADA deficiency is adenosine deaminase deficiency and it causes severe combined immunodeficiency or SCID, SCID. And it requires lifelong treatment through bone marrow transplantation. But with the approach of gene therapy, the T cells from this 4-year-old girl, her name is Shanti De Silva, it was removed and then functional ADA gene was introduced into the T cells using retroviral vector. Then the modified T cell was again injected back into her body. So what technique did they use? They used gene therapy. So answer here is option B. The agent or toxin which requires registration with the US Department of Agriculture Animal and Plant Health Inspection Service under the Agriculture Bioterrorism Protection Act is. So out of these it is complete genetic code for Epstein-Barr virus that requires registration. Then next one, the approximate respiratory ID50, that is number of organisms required to produce infection in half of individuals exposed for Mycobacterium tuberculosis is fewer than 10. Okay, and what does this 10 mean? It means that inhaling fewer than 10 Mycobacterium tuberculosis bacteria can potentially cause infection. That is what it means. Then next one. If every observation in a data set is increased by a constant quantity, then the coefficient of variation of the resulting set of values will be. So CV is equal to mean divided, it is not mean, it is It is standard deviation, sigma divided by mean into 100. That is coefficient of variance. Now the data set, observation in the data set is increased by a constant quantity. That means your mean is going to increase. Now mean increases means CV will decrease because it is inversely proportional. CV will decrease. But standard deviation is not going to change when you increase data set observations. Okay. Standard deviation will remain the same. Only mean will change. So the answer here will be less than the CV of the original data set. Answer will be option A. Then next one. Which of the following statement is incorrect regarding trademark? It may be word, signature, name, device, label, numeral or a combination of colors. That is correct. It prevents imitation of the offering. Yes, you are not allowed to imitate trademark. Registration of trademark is mandatory in India. It is not mandatory, but it is recommended in India. So that is the incorrect statement and that's your answer. The term of registration is 10 years. That's also correct. Now next one. A baker is interested in whether there is a difference in the mean deliciousness rating that customers give to low sugar option of a cookie and to a high sugar option of a cookie, and whether the difference in mean rating for the low and high sugar option depend on the age group of the customer, that is child versus adult. What test should the baker use to examine this question? So here there are two independent variables. One is sugar level, whether it is low sugar or high sugar, and the other is age group, whether it is child or adult. So here two-way ANOVA will be suitable. Answer is option C. Two sample t-test for a difference in mean. It is suitable only for comparison of two groups, that is low sugar versus high sugar, but it will not take age group into consideration. Then B also, it will examine only low sugar and high sugar levels, but it will not take age group into consideration. Then chi-square, it is used for nominal data, not for continuous data. Now this data, whatever you have here is low sugar, high sugar with age group of customers is a continuous data. So chi-square also cannot be used. So the answer here is option C. Then next one. The first product that got geographical indication tag in India. Now what is this geographical indication tag? It is a tag that is used to, or I can say it is a sign that is used to tag the products that have specific geographical origin. Okay, such as these examples: Katrani rice, Kashmiri saffron. It is there in Kashmir only. Kashmiri saffron has got specific qualities, unique qualities. It has got its own characteristic because of its origin. Then Darjeeling tea, it is from Darjeeling. Then Sanganeri hand block print, that is also from that place. So all these things, they are geographical indications. Okay, but they have asked the first product that got geographical indication tag in India was Darjeeling tea, that is in the year 2004. Then next one. When a string is used for numerical computation, Perl converts it into, so it converts it into number. Answer is option B. Then which among the following are closely associated with IPR, that is intellectual property rights? TRIPS. TRIPS stands for Agreement on Trade Related Aspects of Intellectual Property Rights. So A is the answer. What is TRIMS? TRIMS is Trade Related Investment Measures. It deals with investment, not with IPR. Then GATT, GATS, it is General Agreement on Trade in Services. It focuses on service sector trade, not on IPR. MFN is Most Favored Nation. So that is also not specific to IPR. So the answer here is option A. Then next one. Company A produces 10%, Company B produces 20% and C produces 5% defective products. If choosing a company is an equally likely event, then find the probability that the product chosen is defective. Now, since choosing a company is an equally likely event, each company has 1/3 chance of being chosen. So probability of defective product is 1/3 into 0.1. 0.1 I got from 10%. 10 divided by 100, 0.1. 1/3 into 0.2, that is 20%. So 20 divided by 100, 0.2. Plus 1/3 into 0.05, that is from 5% divided by 100. Now take 1/3 out. 0.1 plus 0.2 plus 0.005. You will get 1/3 plus 0.35, that is 1/0.12. So the answer here is option D. Then next one. The extensions for Windows sound file is option A .wave. The computer language used in artificial intelligence is Prolog, option D. Reliefs in action for patent infringement are provided under section 108. Then which of the following is not a bioinformatics software? Amphora, it is a platform, web-based platform for genome assembly, annotation and analysis. Then Embos, it is a collection of open source tools for sequence analysis, alignment and database management. Then ProSite, it is a database, not a software, of protein families, domains, functional sites, etc. GRAIL is a gene prediction program. So the answer here is option C, ProSite. They have asked which is not a bioinformatics software. Then UGC CARE has been set up for promoting quality research, yes. Academic integrity, yes. Publication ethics, yes. Inclusion and access, international collaborative research, no. So 1, 2, 3 only, answer is option B. Then next one, plagiarism in research, that is copying. So copying unscrupulously and making use of it, answer is option B. Identify the correct sequencing of the following steps involved in DNA fingerprinting. So first will be isolation of DNA, that is not given here. Okay, digestion with restriction enzyme. Yes, after isolation of the DNA, you digest it with the restriction enzymes. Then you electrophorese the DNA fragments. Then you blot the DNA fragments onto nitrocellulose membrane. Then hybridization with the DNA probe. And finally autoradiography. So 3, 1, 5, 2, 4. 3, 1, 5, 2, 4 is your answer B. Then dead zones. So dead zones, they refer to areas which have extremely low oxygen concentration, that is hypoxic conditions. And because of this low oxygen levels, it will be difficult for most of the life to survive there. So what are dead zones? The deepest zones of the Mariana Trench where living organisms are not found. No. Neritic zones that are devoid of sunlight at extreme end of continental shelf? No. Hydrothermal vents having extreme temperature, still organisms grow there. Extremely hypoxic zones. Yes, that is correct. Organisms are not found there. Due to substantial eutrophication. Yes, and what is eutrophication? It is discharge of these fertilizer sewage from industries into the water, which will result in excessive algal growth and depletion of the dissolved oxygen. So it will also create a hypoxic zone, and because of this hypoxic zone there will be no life surviving there. So answer is option D. Then as per Central Pollution Control Board norms, the maximum permissible limit of 3 days BOD at 27 degrees in wastewater effluent when discharged into an inland river or stream is. So what is the maximum permissible limit that you can discharge effluents into inland river and stream? So the maximum permissible limit of BOD is 30 mg per liter. Now 100 mg per liter, it is excessive pollution and harmful to aquatic life, so that they will not allow. 50 mg per liter is high pollution, potentially harmful to aquatic life, so that also not allowed. 10 is very low pollution and generally considered safe, but they have asked here maximum permissible limit, so it is 30. Then which of the following is a secondary pollutant? So secondary pollutant, they are formed from primary pollutants, and when these primary pollutants, they react with substances in the atmosphere and give rise to secondary pollutants. Out of these, smog is a secondary pollutant, and it is formed through chemical reaction between primary pollutants, which are nitrogen oxide, volatile organic compounds and ozone. Whereas carbon monoxide, fly ash, freon, they are primary pollutants. So that's it. Thank you for watching my channel. Please do like, share and subscribe if you are new to my channel. If you want still previous year question papers of Kerala SET Biotechnology being discussed, you can go to my channel Omni Bio Entrance. In the playlist C for Kerala SET exams, in that you will find. Thank you.